In this video, we will go over practical issues involved in designing a genetic screen. There are many genetic model organisms available to us now. They can be prokaryotic bacteria, or bacteriophage, or viruses. For eukaryotic organisms, you can choose to work with the single-cell model, the yeast, or multicellular invertebrate model like C. elegans and Drosophila. You can use Arabidopsis as a plant model, and zebrafish and mouse are commonly used vertebrate models. Of course, you can choose to work with embryonic stem cells or induced pluripotent stem cells if they suit your need. Now I want you to take a moment to think about what makes a good model organism for genetic screens. The answer is F. You want your model organism to have a short life cycle and can produce a large number of progeny. This is important because you don't want to wait for a long time to do one experiment. You also want the model organism to have the structure or function that are relevant to your question. For example, if you want to study heart development, then the yeast may not be your first choice because they are single-cell organisms which do not have a heart. In the rest of the video, I will use the zebrafish heart as an example to help you design your own genetic screens. So what is zebrafish? The zebrafish is a small tropical fish. We can grow tens of thousands of them in a small laboratory setting. They are vertebrates and they have a heart. You may ask that the fish looks very different from other animals. Can it help us understand how human hearts develop? It turns out that even though the adult animals may look very differently, at the embryonic stage, all vertebrates look very similar to each other, and the embryos follow very similar morphologic processes. All vertebrate hearts develop from the fusion of the bilaterally positioned precursor cells. After the formation of the heart tube, cardiac chambers continue to differentiate and mature. The advantage of using zebrafish as a genetic model is that they produce large number of progeny, and their development is very fast. Within 24 hours, a fertilized egg can develop into an embryo with a beating heart that can support the circulation throughout the body. More importantly, the zebrafish embryos are transparent and develop in water. This allowed easy access to examine the phenotypes. Using transgenic technology, we can label specific cell types with fluorescent proteins like GFP. Shown in this slide are transgenic fish embryo in which GFP expression is driven by a vessel-specific promoter KDRL, so that you can observe how the vascular system develop real-time in live embryos. In this movie, we show an embryo where we use KDRL promoter to drive GFP expression in all vessel cells and GETA promoter to drive the red fluorescent protein expression in blood cells. You can then monitor the heartbeat and blood circulation throughout the body. Now, if you accept that fish is an appropriate model to study heart development, how do we create mutants with defects in heart formation or function? Just like what you learned in the genetic class, you can mutagenize the fish genome using mutagens. You can use chemical mutagen like ENU to induce point mutations. You can use radiation treatment to create a large deletion or rearrangement of chromosomes. You can also use retrovirus or transposons to create insertions. Furthermore, with the new molecular tools and the completely sequenced genome, you can systematically knock down or knock out genes by morpholino-modified antisense oligonucleotides or CRISPR-based genome editing technology. In the late 1990s, Jannie Nusslein-Volhard led another large-scale genetic screen, this time in zebrafish. She designed a 3-generation breeding strategy to identify recessive mutations with defects in zebrafish development. Among these are a group of mutants with defects in heart formation or function. Now let us take a moment to reflect the reasoning behind this 3-generation breeding strategy. In this screen, researchers mutagenized the fish genome by soaking male fish in ENU solution to induce point mutations. They carefully titrate the concentration of ENU and use the condition that will induce 1 mutation per genome. This means that each sperm this male fish produces carries a unique mutation. If we cross the ENU-treated male fish to a wild-type female fish, each one of their progeny will inherit a unique mutant allele from their ENU-treated father. Now, if we screen the F1 fish, what types of mutants do you expect to find? The answer is A, dominant traits. Zebrafish is diploid, which means that they have 2 alleles of each gene. In the case of the mutated genes, the wild-type allele is inherited from the wild-type mother and the mutated allele from the ENU-treated father. In order for the F1 fish to show the mutant phenotype, the mutation must be a dominant trait. For recessive mutations, we need to breed them to homozygosity to see the phenotype. What we need to do is to cross the heterozygous F1 fish to a wild-type fish. Half of their progeny will be wild-type, and the other half will inherit the mutant allele. Now, if we randomly cross F2 fish to each other, we expect that 1 out of 4 crosses will be heterozygous male crossed to a heterozygous female, and 1 out of 4 of their progeny will be genotypically homozygous for the mutant allele. We can then examine their phenotype to determine whether this gene is involved in the regulation of heart development. At the end of the screen, they identified hundreds of mutants with specific defects in heart morphology or function. For example, we have mutations that prevent the fusion of the cardiac precursor cells, resulting in the development of 2 beating hearts in the mutant embryo. We also have mutations that interfere with cardiac chamber formation or the growth of cardiac tissue or the contractility or the rhythm of the heartbeat. In the screen, we found many mutants that have 2 beating hearts. Now consider the following statement and choose the correct answer. The correct answer is D. We need to do a complementation test to determine how many genes we are looking at. The concept of the complementation test is the same as what you learned in Benser's R2 experiments. You will need to cross 2 mutant heterozygotes to each other. If all their progeny show wild-type phenotype, that means they complement each other and they represent different genes. If a quarter of their progeny show mutant phenotype, in this case, have 2 beating hearts, that means they fail to complement each other. These mutations represent different alleles of the same genes. Now we have our mutants, and we perform complementation tests to figure out whether they represent different genes or same gene, different alleles. We can consider the next question, that is, identifying the gene that is responsible for this mutant phenotype. The first step is to map the mutation. Once you place the mutation on a specific site of a chromosome, positional cloning will help you identify candidate genes. Now, the genomes of most model organisms are completely sequenced. You can rely on genomic information and informatic approaches to narrow down the candidate genes and perform functional analysis. How do we map the mutations? We need to investigate the linkage relationship between the mutation and genetic markers. Earlier this quarter, you have learned about polymorphism and molecular markers like RFLP, SNP, and microsatellites. In zebrafish, the most commonly used markers are microsatellite markers. In the genome, there are many repeats like CAs, TAs, and GTs. In different genetic backgrounds, these repeats may vary in numbers. For example, in one strand, the CAs may repeat 50 times, but in another strand, only 40 times. If you use PCR primers flanking these tandem repeats, you will generate bands with different sizes. This is the base of the polymorphism. Let's assume that you generate your mutation in the zebrafish AB strand. To map the mutation, you need to cross the heterozygous individual to a fish from a polymorphic background, for example, the wick strand. The F1 fish will have one allele inherited from the AB parent and the other allele inherited from the wick parent. Now, if you cross F1 fish to each other, the F2 fish could be homozygous for wick or homozygous for AB or heterozygous for wick and AB. How would this translate to polymorphism and linkage, and how can this help us map the mutations? What you need to do is to collect genomic DNAs from parental F1 and F2 fish, then perform PCR analysis using the primers flanking a specific genomic region that has the CA repeats. Let us assume that you have a polymorphic marker that is linked to your mutation. This pair of PCR primers flank a genomic region that has 50 CA repeats in the AB strand and 20 CA repeats in the wick strand. The PCR fragment from the parental AB and wick fish will show polymorphism in their size. The PCR fragment from the AB strand is larger than the PCR fragment from the wick strand, so it runs slower. F1 individuals are heterozygous, therefore you will detect two bands. When you examine the PCR fragment of the F2 individuals, you need to pay attention to their phenotypes. Because zebrafish embryos are transparent, you can easily observe the phenotypes. Those F2 individuals with wild-type phenotype could be genotypically wild-type, which means homozygous for the wick allele, or heterozygous, which means carrying one AB allele and one wick allele. Their PCR results will be either showing only the wick band or showing both AB and wick bands. Those embryos with two beating hearts must be homozygous for the mutation because this is a recessive trait. If the CA repeat marker is very closely linked to the mutation, there will be no recombination between the marker and the mutation, and you will detect the AB band only. If they are far apart, recombination may occur between the marker and the mutation. In that case, you will see both wick and AB bands. By examining the PCR results of the F2 mutant embryos, you will be able to calculate the genetic distance between the mutation and any given markers. The calculation is the same as what you learned before. The genetic distance equals to the number of recombination events over the number of meiosis examined. For a diploid individual, each embryo represents two meiosis events, one during father's gamete formation and the other during mother's gamete formation. Therefore, the genetic distance will be the number of recombination events over 2 times the number of F2 mutant embryos examined. If the mutation and the microsatellite marker are on different chromosomes, they will segregate independently, so the calculated recombination frequency will be about 50%. For those markers that are linked to the mutation, the recombination frequency will be below 50%. The closer they are, the less recombination events you will detect. Now we will look at this example. You examined 100 zebrafish mutant embryo and detected 50 recombination events between a microsatellite marker and your mutation. Is this marker linked to the mutation? If so, how far apart are they? The answer is C. They are linked. 50 recombination events detected in 100 mutant embryos means 50 recombination events out of 200 meiosis equals to 25% equals to 25 centimorgan. To map mutations in the lab, we usually start by setting up a panel of 96 PCR reactions. Zebrafish has 25 chromosomes. We choose approximately 4 markers per chromosome. That means a plate of 96-well PCR reaction can give you a rough idea where the mutation is in the zebrafish genome. Since the genome is sequenced, once you map the mutation, you can check the genomic region for candidate genes. Consider this situation. From linkage analysis, you found that gene X is tightly linked to the cardiac bifida mutant A. Which of the following will convince you that cardiac bifida is caused by the mutation in gene X? The answer is D, all of the above. A nonsense mutation in gene X indicates that mutant protein of gene X may not be functional. If indeed gene X protein is needed for cardiac fusion, adding wild-type gene X back to the mutant embryo should allow the bilateral primordia to fuse and form the heart tube. Conversely, knocking out gene X in wild-type embryos should inhibit cardiac fusion, resulting in the development of 2 beating hearts. To conclude, now you watch these 2 parts video, you should be able to discuss how model organism and genetic screens can help you identify critical genes in the biological processes of your interest. You should be able to describe the regulatory mechanisms that establish the body plan in the fruit fly. You should also be able to select a suitable model organism and design a genetic strategy to study your favorite biological questions.